Hey there, and welcome to this explainer. If you've ever felt like you're totally drowning in an absolute sea of academic papers, data sets, and patents, trust me, you are definitely not alone. Today, we're basically putting together a survival guide. We're going to figure out how to navigate the dark, overwhelming void of unstructured information using some truly brilliant tools. Because, let's be real, in today's knowledge ecosystem, finding the right information isn't just about grinding harder. It's about knowing exactly which tool to pull from your tool belt for the job.
Okay, let's just quickly look at our roadmap. We're going to hit the research data avalanche, then move on to broad platforms, specialized tools, structured versus unstructured data, and we'll wrap up with the fair golden standard.
All right, part one, the research data avalanche, grasping the massive scale of scholarly output.
So, before we even touch the tools, we really have to wrap our heads around the sheer scale of the environment we're operating in. The modern researcher is up against a volume of global data that is, well, almost incomprehensible. And it's not just the volume, right? It's the crazy complexity and the breakneck speed at which new data drops every single day. Just think about this number for a second. 3.3 billion. That is the number of citation links currently tracked by just the Web of Science platform alone. 3.3 billion connections between ideas, papers, and discoveries. If you tried to navigate that manually, you'd literally be lost in seconds. This staggering figure really highlights why we desperately need computational systems to do the heavy lifting and navigate this massive ecosystem for us.
Moving on to Section 2, broad platforms, the megastores. Basically, the Swiss Army knives of research.
So when you're faced with that 3.3 billion link avalanche and you need to start a literature review, you usually reach for your multi-purpose tools first, the megastores. Think Scopus with its 100 million plus records, or Web of Science, which covers hundreds of different subject categories. These platforms are phenomenal for giving you that immense multidisciplinary coverage. They let you cast a really wide net. Like you can see how a hyper-specific concept in physics might suddenly apply over in biology or engineering. They're incredible for spotting the interconnectedness of science. Plus, their access models run the gamut from hefty subscriptions all the way to free options like Google Scholar. But what happens when your trusty Swiss Army knife just isn't precise enough for the job?
That brings us to Section 3, specialized tools, the scalpels, domain-specific depth and precision.
Because here's the thing, once you cast that super wide net, you almost always hit this massive wall of noise. Broad search terms just start spitting back way too much irrelevant data, and that is exactly when you drop the Swiss Army knife and pick up a scalpel. This comparison is absolutely crucial for modern researchers. While you might use a broad platform like Web of Science to track huge trends across, say, 254 different subject categories, you'd use a highly specialized tool like MatKG to surgically extract exact quantitative properties. MatKG is actually the largest knowledge graph in material science. It doesn't just do basic keyword searches, it literally maps out structure-property relationships at the embedding level. So a broad platform might tell you, hey, these two papers use the exact same word, but a specialized tool, it tells you exactly how a specific synthetic iron oxide behaves in the real world. It's all about depth over breadth.
And there are other scalpels out there that offer some incredibly niche utility. Doing patent analytics, a standard search engine is going to fail you. But a tool like the Lens lets you do bulk downloads of 50,000 records at a time, complete with claims and abstracts. Or maybe you need genetic sequence listings. PatentScope is an absolute game changer there, letting you pull massive gigabyte-sized data dumps right over FTP. And for nucleotide sequences, GenBank offers highly curated industrial-scale data. Basically, these niche tools ensure you don't miss that one critical patent or sequence that a broad search would completely gloss over.
All right, Section 4, structured versus unstructured data, formatting the foundation of science.
Now, we've talked a whole lot about where to search for information. But actually, scratch that. Finding an article is only half the battle. Using the raw data behind it is a completely different beast. We need to shift gears and look at how that data is actually formatted, stored, and shared. And that brings us to a really brilliant point by Andrew Houghton, managing editor at Scientific Data. He puts it perfectly. We would like authors to put their data in the most appropriate place for that data, because science is moving away from this old-school cottage industry model, where isolated labs just hoard data on local hard drives. We're moving toward global, industrial-scale collaboration, and in this new reality, where you put your data is just as critical as how you discover it.
Which leads me to ask you directly. So, where exactly does your data belong? Think about your own day-to-day workflow, or what your organization puts out. Are you generating super standardized sequences? Or are you creating highly unique spreadsheets and weird video files? Because honestly, your answer totally dictates the tool you need to use. Basically, there are two main paths for your data. If your data is highly unique or just kind of doesn't fit a standard mold, like maybe it's a video of a behavioral study or some totally custom spreadsheet, it belongs in an unstructured repository. Think Figshare or Dryad. These guys are super flexible. They're essentially where everything else goes. But on the flip side, if you're part of an industrial-scale science project, like, say, an omics field that's churning out nucleotide sequences, your data belongs in a structured repository, like GenBank. Structured repositories enforce really strict formats, which makes the data highly curated and, most importantly, easily readable by computers.
Which leads us nicely into Section 5, FAIR, the golden standard, unifying the modern knowledge ecosystem.
So to keep all these megastores, precise scalpels, and various repositories from just completely descending into total chaos, the scientific community leans heavily on modern data stewardship, and that is perfectly represented by the Go FAIR initiative. These principles were published back in 2016 for one very specific reason. The sheer volume of data has grown so outrageously huge that we humans just can't manage it alone anymore. We have to rely on computational systems to help us navigate this whole ecosystem.
But for a computer to actually help us, the data has to follow 4 golden rules. First, it needs to be findable. That means having globally unique, persistent identifiers and really rich metadata. Second, accessible. Once you find it, you need to be able to retrieve it using standard, open protocols. Third is interoperable. It has to use shared, broadly applicable languages so entirely different systems can actually talk to each other. And finally, reusable. It needs crystal clear usage licenses and detailed provenance so other researchers can confidently build upon your work. The whole point of these principles is to ensure that data doesn't just get dumped into that dark void we talked about earlier. Instead, it's actively structured so machines can use it with minimal human intervention.
To wrap the whole shebang up, I want to leave you with a final thought. You now know the tools, right? From the massive, multidisciplinary megastores to the hyper-precise scalpels. You see exactly why researchers have to carefully choose between broad platforms like Web of Science and specialized tools for true depth. And you understand the critical choice between structured and unstructured repositories. So, as you go out and contribute to this massive ecosystem, ask yourself, will your next data set just get lost in the void? Or will it be a findable, interoperable tool for the future? Choose your tool belt wisely.